Hi, I'm Steven from Ghent University and welcome to my poster about investigating a novel Artemis mutation to establish a valid in vitro model for defining genotype-phenotype correlations in SCID. SCID, or severe combined immunodeficiency, is a life-threatening immune disease caused by an increasing number of genetic defects. These patients suffer from opportunistic infections as a result of an abrogation of T lymphocyte development and/or function, in which B and NK cell development and/or function might be affected as well. Therefore, SCID is characterized by a heterogeneous spectrum of both genotypes and phenotypes, which often complicates the diagnosis of SCID. When we encounter a patient in the clinic with a SCID phenotype, we perform targeted next-generation sequencing using different panels to reveal the underlying genetic defect. Here we can identify new variants in known SCID genes, but on the other hand, variants in candidate genes can be identified that are not yet known to cause SCID. Next, we validate the causality of these SCID genes in our in vitro disease model by isolating hematopoietic precursors from peripheral blood and differentiate these cells towards T, B, NK, and myeloid cells on different feeder layers in the presence of lineage-specific cytokines. In this way, we're able to identify a stage-specific arrest during the development of these progenitors by weekly follow-up using flow cytometry for stage-specific markers. Eventually, we can correlate the in vitro phenotype to the genotype that was found initially to confirm the causality of the variant in these SCID genes. This has two important implications. From a fundamental perspective, we can try to uncover the role of genes that are important to drive immune cell development, with the addition of molecular studies to reveal the mechanism that caused the developmental arrest. On the other hand, this approach can help to improve diagnosis, treatment, and follow-up in the clinic, and thus has a translational impact. Thank you for your attention.